Okay, so in this video, we're going to talk about how proteins fold up. So there are 4 overall structures to how a protein folds. So like 4 steps. And it doesn't like happen first and then pause and then number 2 and pause. It's like a fluid movement of the folding up of this polypeptide. So let's go ahead and look at the first sequence. It's basically called the primary structure. So that's the first step in how a protein folds. And this structure is based on the sequence or order and length of amino acids. So the primary structure is basically the building of the polypeptide, and it's based on peptide bonds. So I actually have an earlier video that shows just how a peptide bond is formed between 2 amino acids. And so as we link those amino acids together using peptide bonds in the process of dehydration synthesis, that forms the primary structure. Now, each protein is a unique length of amino acids and a different sequence or order of amino acids. And so we have a huge variety of proteins on Earth, and their primary structures or their sequence of amino acids do vary from one another. Okay, so let's go ahead and see then the secondary structure. Once we form this polypeptide, which by the way happens in ribosomes during protein synthesis, it needs to begin to fold up. So this is where we have our secondary structure. Now this was really hard to show in animation. So I want you to imagine here that primary structure, the polypeptide, like folding around is kind of what I was hoping to demonstrate here. And as this polypeptide, which I just have a string here on my desk, but imagine like it begins to like fold up is kind of what we're looking at. And so here, this secondary structure is actually based on hydrogen bonds. So hydrogen bonds, there's an attraction between the nitrogen of an amino group and an oxygen from a carboxyl group of a different amino acid. So hydrogen bonds begin to form between like the backbone of the polypeptide, and it actually causes, it actually causes the polypeptide to fold up into 2 distinct shapes. So we have what's called the alpha helix, which causes like a spiral shape in that polypeptide. And then we also have a beta pleated sheet, which is more of like an accordion or folds in that polypeptide. But these like white lines between them are the hydrogen bonds that are responsible for the secondary structure or the second step in protein folding. Now, let's go ahead and talk about the third step. So this is called the tertiary structure. And I want to point out that each amino acid, R group, has unique chemical properties or chemical traits or characteristics. Some R groups are positively charged, some are nonpolar, some are negatively charged, some have sulfur, and some are polar. So as we have this polypeptide that is now folded up into an alpha helix or a beta pleated sheet, now these R groups are going to begin to interact with each other, beginning that 3-dimensional shape of the protein. So here I'm going to just use a squiggly line to represent our polypeptide, and then now those R groups begin to interact or with each other. So that could mean like repel or attract to each other. So if we think about proteins, proteins are found in cells and outside of cells, but no matter if they're found in the cytoplasm or inside an organelle or sent out of the cell like insulin is, these proteins are surrounded in water. And nonpolar R groups are going to repel water. So as a polypeptide folds up, you're going to find those nonpolar R groups are going to be located towards the center of the polypeptide or of the 3-dimensional shape of the protein. You're going to find polar amino acids interacting and attracting to the watery or aqueous environment. Now, if we recall, some amino acids have positive charges, or the R groups, and some R groups have negative charges. So they are able to form ionic bonds, which is an attraction between a negative and a positively charged molecule. So we have ionic bonds begin to form between R groups, which help stabilize and build that shape. We also have So there's still some hydrogen bonds left over, or still there from the secondary structure, but new hydrogen bonds can form. And then 2 out of the 20 amino acids have sulfurs. So when 2 sulfurs come together, they actually can form a disulfide bridge, which is actually a covalent bond, which really does stabilize this tertiary structure. So when I look at the overall takeaway of this third step of protein folding, this tertiary structure is based on how these R groups interact with each other, whether they're attracted to each other like nonpolar and nonpolar, or whether they're repelling each other like a polar and a nonpolar would repel. And so, or whether they're bonding ionically between a positive and a negative, or covalently between a sulfur and a sulfur, or maybe it's a hydrogen bonds between 2 R groups or 2 amino acids. And so all of these interactions develop the tertiary structure of this polypeptide. Okay, so now the quaternary structure is composed of multiple polypeptides joining together to determine this protein's overall shape. For example, hemoglobin is the protein in our red blood cells that carries oxygen, and hemoglobin actually has 4 polypeptides together. And so similar interactions that we saw in the third step will also hold the polypeptides together. So like attractions between the polypeptides will kind of group them. And so when we think about the wide diversity of proteins on Earth, it really comes down to that sequence of the first step, the sequence of amino acids and what R groups they have. And then those R groups will really determine the shape of the protein. And it's super important to remember that the shape of the protein is how it does its job. So shape determines function. If there's a mutation in the DNA that changes leads to a change in amino acid, it could change the R group properties, which then would change this tertiary structure. So mutations is a whole nother topic, but when there's changes in the order of the amino acid, it could lead to changes in this tertiary and quaternary structures, which lead to a protein that either doesn't work or works differently, etc. All right, so great job.